Hello, good morning, everyone. My name is Sreenivasulu Kurukuti from University of Hyderabad. Today, I'll be discussing with you about human genome and then epigenome regulation. How human genome is structurally and then functionally organized, and how the epigenome within the genome is, is able to confer the cell type specific gene expression patterns that what you see in human body. So just begin, life of all of organisms starts with a single cell zygote. As we know, zygote is a diploid cell, which is considered as a start point of any kind of a life of an organism, which is basically formed by the union of the act of fertilization by haploid oocyte and then haploid sperm. And then this diploid zygote eventually, by means of cell proliferation and then differentiation, gradually give rise to morula and then into blastula and then gradually develops into an embryo and then grows to an adult organism. Depending upon the genotype, then it could be a male or it could be a female. Again, these adult organisms, when they attain puberty, they produce germ cells, like for example, females oocytes and then male sperms, thus fulfilling the life cycle. Then here, if you look here, the one of the fundamental questions in human biology is that how come a single cell zygote has the capacity to give rise to entire organism? Because organisms are made up of millions and billions of cell types, and then from how come that single cell can give rise to such a plethora of cell types with a diversity in their function? If you look closely into the early embryonic development, and soon after the morula and then the blastula formation, the cells that are present at the interior of this particular blastocyst eventually give rise to entire embryo, and these the singular trophoectoderm that bears within the blastocyst that eventually give rise to the placenta, which connects the mother to the fetus. And these cells, which are present at the interior of the cell, the blastocyst stage, they can be excised and they can be propagated in vitro, and then they are considered as embryonic stem cell. And these embryonic stem cells can able to differentiate into multiple cell types of human body. And then the question comes here that how come these stem cells, embryonic stem cells, which are present at the blastocyst stage, can give rise to such a diversified cell types, in spite of the fact that all these cells that are constituting the human body, they all are having identical genome, but every cell is unique, and then they have its own set of genes being transcribed, one set of proteins being translated, and then one set of metabolites are present in each and every cell, and they constitute a unique function for each and every cell. Then one of the fundamental questions here is that how come, in spite of having identical genome in all these cell types, each and every cell has a unique gene expression program? For example, here in this particular cartoon, it shows that the cells that are excised from early blastocyst stage, they can be propagated in vitro, and then they can be directed to differentiate into multiple cell types such as muscle cells, neuronal cell, and then intestinal cell. And then, as we know, that entire new branch of science, that is regenerative medicine, takes advantage of this particular breakthrough in this technology of in vitro differentiation of embryonic stem cell to a defined cell types. And then, in order to understand how that, in spite of having an identical genome, that cells, how come that they transcribe different set of genes? If you take a human genome, our genome is present in a specialized compartment within the cells. This is called a nucleus. And then in this particular photograph, the nucleus is stained with a dye called DAPI. Hence, when you observe these cells under fluorescent microscope, you can see them as a blue color. Thus, this deoxyribonucleic acid within the nucleus is as I said, it is present within the cell nucleus, the spherical geometry. And then, if you look into this particular DNA, it is not a continuous thread of DNA that is present within the cell nucleus. It is discontinuous, and then every chromosome or every DNA that what you see, all these DNA molecules do not exist as a naked DNA. They are always in association with a strong group of proteins, and then they form a chromatin. And then each and every DNA fragment, along with this histone group of protein, forms a chromatin. And these chromatin belong, we refer them as a depending upon the length, we name them as a chromosome number 1, 2, 3, 4, and so on. That means our typical human cell nucleus harbors fragmented genome, and then which appears in the form of a chromosomes, which can be clearly seen during certain stages of cell cycle. As you can see here, that each and every chromosomal DNA by condensation appears like chromosomes during certain stage of cell cycle. So here I would like to say that so the chromatin, that the DNA that what we have in the cell, is highly dynamic. During interphase it is highly diffused, and during certain stages of cell cycle it is highly condensed. It can be duplicated, it can be condensed, and then they can be segregated, and that's how the cell cycle progresses. And then if you go much deep into the individual cell and then a nucleus, if you take a cross section of the cell nucleus and try to observe under electron microscope, you see there are some kind of differences within the way the genetic material organized within the cell nucleus. As you can see here, the nuclear membrane, double layered nuclear membrane with some kind of nuclear pore complexes. And then if you see the chromatin within the cell nucleus, you see that there are two distinct regions that can be seen very well evident. One is electron dense euchromatin. As you can see, it is more or less restricted to the nuclear periphery, and then electron poor, that is euchromatin, which is present in the nuclear interior. Now we know, we will establish fact that in a given particular cell type or in a particular differentiated cell, all the genes, all the chromatin which is not transcribed or which is not useful, it is heterochromatinized, very highly condensed, and then they are more or less pushed towards the nuclear periphery, whereas the transcriptionally active genome, which is loosely packaged, and then they are seen at the nuclear interior. That means there is a compartmentalization of gene activity. More or less, gene activity can be seen at the nuclear interior, and then gene silencing one can see at the nuclear periphery. And then within this context of highly heterogeneous chromatin that what you see under electron micrograph, if you try to see what are the way exactly genes are present, we know that, for example, in a typical human cell nucleus, if you take any particular chromosome harbors multiple genes, and then every gene is regulated by a group of sequences called either enhancers or silencers, and these enhancers or silencers are present far away from the gene body. Some instances even regulatory elements such as enhancer for a given gene can be present in different chromosomes. So, but three-dimensionally now it is well explored that when genes are transcriptionally active, these cognate enhancers, which are present far away from the gene body, they are seen very physically close to the promoter. Thus, it triggers a transcriptional activation. Similarly, or for the same case for some kind of genes where the regulatory elements are present in far away. And in this context, very important to understand how that this nuclear organization of genome influences such a precise and the crosstalk between these regulatory elements and the genes, which are maybe present within the vicinity or sometimes very far distances. In that context, it is very important for us to understand how that genes, what we know, how that chromosomes, what we know, and then how actually they are seen within the telomere space of the nucleus. And at the same time, having said that, all cells are having identical genome, and then but every cell type has a unique gene expression pattern. That means, for example, in case of human genome, actually codes for nearly 30,000 genes, but all genes are physically present. All the DNA that is present, physically present in every cell, but not all genes are transcribed in a given cell type. Each and every cell transcribes a unique set of genes. That means gene regulation is regulated. So in the context, how genes are regulated, how a particular cell knows that these are the X or Y genes, which needs to be transcribed in a cell type specific manner. So in the context, now we know that these genes are regulated at multiple levels in a given cell type. Cell type. So in the context, gene signatures for a given cell type are actually predetermined even at the pre-transcriptional state, before the cell is being formed, either liver cell or muscle cell, whether either before a cell is being formed, and then it already decides which genes are going to be transcribed in a given cell type. This kind of regulation is at a pre-transcriptional. In that context, replication timing, chromatin accessibility, chromatin condensation, chromatin supracoiling, and then three-dimensional chromatin organization plays a very pivotal fundamental role in deciding which genes are going to be transcriptionally active in a cell that is just formed. And then further, even once the cell is formed, for again the genes are further regulated at transcription level. That again depending upon the DNA methylation, and then presence or absence of a few cell type specific transcription factors, and then histone modifications, and then chromatin remodeling. They play at the transcription level fundamental role in whether or not to transcribe a given gene. Further, genes are further regulated at post-transcription level. That happens by means of alternative splicing, microRNAs, long non-coding RNAs, RNA modifications, RNA binding proteins, mRNA stability and the degradation. These things at post-transcription level play a fundamental role in establishing a cell-type specific gene expression patterns. Not only these three, but again, even genes are regulated at a translation level in terms of specialized ribosomes, and then in terms of initiation, elongation, and termination during translation, and then microproteins, they do establish a cell type specific gene expression pattern. Further, beyond translation, there are post translational modes by which genes also can be regulated, gene products can be also be regulated, such as protein post translation modifications, and then protein stability, protein degradation. All these five modes of gene regulation will ensure that a given cell type has a unique gene expression pattern. Further, in spite of the fact that in our in humans, nearly 75 different, 170 different five different kinds of cell types are present, and then these cells, in a day-to-day fashion, they will be encountering with the multiple signals. For example, hormonal signals, or heat stress, or some kind of stress signals. All these signals, by means of receptor-mediated cell signaling, or it is a cytoplasmic located receptor-based signaling. These signals are transduced to the genome or non-genome fashion. Thus, they differentially transcribe multiple genes. That means even extracellular signals can serve as some kind of epigenetic signals, so which will formulate in such a way that genes can be differentially transcribed. Further, recently, it has been shown that although for example, any kind of cell type, if they do not have any kind of a products, but it can borrow those genes or gene products from different cells, and that is achieved through this one of the very recent phenomenon called by means of exosomes. Here, in a given cell type, exosomes are formed, which are nothing but which is a membrane bound small organelles, which has a defined microRNAs, defined transcripts, defined proteins. So, like a cargo, they can be packaged and they can be released into the away from the cell in the extracellular space. And these cargoes can go and they travel through even the bloodstream, and they can go to distal places in the specialized cells or special cells, where they can go and then they can fuse with those cells. Thus, they can enter into the new cells, and then they can change the gene expression pattern. That's how genes genetically and then epigenetically, by means of various modes of gene regulation, they will ensure that cells has a unique transcription program. Thus, they do their functions where they are within our different organ systems. Okay, broadly, to categorize the genes in a cell type specific manner, what are the features, what are structural features of a genes that will define a transcription status? For example, in this particular slide, the left half shows the structural features reminiscent of a gene which is silent, and then structural feature, right side, the structural features that are reminiscent of a genes which are transcriptionally active. So, in that context, uh, uh, the silent genes in a given cell type are known to have a heavily methylated, whereas the active genes are known to be heavily demethylated. At the same time, nucleosome positioning is very ordered fashion is seen within the silent genes, whereas the nucleosome positioning is highly disordered in case of genes which are heavily being transcribed. Further, the histones that constitute the nucleosomes, they undergo post-translation modifications, and these post-translation modifications, which are special to specific active genes and then silent genes. In that context, H3K9 trimethylation, H3K27 trimethylation, histone H3 lysine 9 undergoes trimethylation. This is the hallmark for all genes, all chromatin regions which are transcriptionally silent, and they have this special histone modification. Further, all the chromatin regions in a given cell type which are heavily transcribed or which are transcribed, they have this kind of unique modification such as H3K4 trimethylation, H3K36 trimethylation. That means there exists some kind of epigenetic code in the form of histone modification that will instruct whether the genes to undergo transcriptional activation or gene silencing. Further, the histone deacetylation is correlated with the gene silencing, and histone acetylation is correlated with gene activity. And then beyond this, and then genes which are silent in a given cell type, they're packaged in a very condensed higher order fashion. Thus, all the chromatin is highly inaccessible to any kind of DNA binding proteins which can bind to a chromatin. Thus, they prevent the binding of DNA binding protein. Thus, these genes, these chromatin regions are kept under silent. Whereas active genes, their chromatin is more or less open, where it can access the DNA binding protein such as RNA polymerase 2. Thus, the genes can be specifically activated. And then at much higher spatial level within the nucleus, the genes which are highly condensed, they are segregated towards the nuclear periphery, constituting some kind of silent borders. Thus, all the genes which are not transcribed, or all the chromatin which are not transcribed in a given cell type, they are heterochromatinized, they are highly condensed, and they are pushed towards the nuclear periphery. Whereas all the chromatin which is transcriptionally active, very loosely bound, and they are seen at the nuclear interior, which can access the transcription apparatus. Thus, genes are kept under active condition. So these are the fundamental, the basic features by which the structural features of the genes defines the transcription status in a cell type specific manner. Further, as I said, the humans are having more than 170 different kinds of cell types, and then all these cells are having more or less identical genome, and then each and every cell has a mother component as well as the father component. Each and every cell has a 23 pairs of chromosomes, and then one set from mother and other set from father. And then all genes are not present in every cell type, but all genes are not transcribed in each and every cell type. Approximately 4 to 6,000 genes are transcribed in a cell type specific manner. And then if you look into the genes that are being transcribed in a given cell type, it could be that sometimes it will be transcribed from both the copies, that is mother copy as well as a father copy. Such kind of genes are called biallelic expressed genes. Sometimes genes are expressed either from a mother copy or a father copy. They're called monolithic gene expression. Or some instances, genes are transcribed in a parent of origin specific manner, and these are called embryonic genes. Apart from these genes, we do have a group subset of genes are called poised genes. That means these genes are not condensed. These genes are not transcribed. These genes are not condensed, but these genes are in open configuration, but they are not transcribed. That means it has a characteristic features of both silent genes as well as active genes. And then one of the characteristic features of these poised genes is that these are the genes with open genome configuration, and then where its promoters are bound with RNA polymerase 2, but they are not transcribed. That means we have a set of genes in our genome. They are not transcribed, but they're already bound with the RNA polymerase 2. They are waiting for some kind of signals in their lifetime, so that as and when appropriate signal comes, the genes which are already loaded with RNA polymerase 2, they can be instantaneously can be transcribed. That depends upon the kind of signal and then activated cofactors. And then thus we have a soldiers which are ready with each and everything, that as and when the appropriate signal comes, and then these subset of genes are specially transcribed in a cell type specific manner in order to combat with any kind of extracellular signals. So likewise, all cells are having identical genome. All cells are having approximately 30,000 genes, but all genes are not transcribed. Only subset of genes are transcribed in a cell type specific manner. But there are some kind of genes which are always transcribed in each and every cell. These are housekeeping genes or ubiquitous genes. Apart from that, there are cell type specific gene expression genes are being transcribed in a cell type specific manner. Okay, so now see, till now we learned about what are the structural features of the active genes and then silent genes. And then if you see those genes, how they're actually present, as I said, this is the electron micrograph of typical mammalian cell nucleus. You can see that heterochromatin regions which are highly electron rich, they are present in the nuclear periphery, and then electron poor regions of the chromatin, which we call it as a euchromatin, they are seen at the nuclear interior. And interestingly, you also find a very big electron dense entity that is seen at the within the nucleus of a typical human nucleus, and this we know is called nucleolus. Nucleolus, I'm going to tell you in my subsequent classes, it is nothing but it is a center where genes which code for ribosomal RNA are transcribed in a single location, and then they form such a big factory-like structure which is dedicated exclusively for transcription and then assembly of ribosomal RNA and then protein, which we call it as a nucleolus. So that means highly silent genes are also condensed, whereas the active genes are decondensed. But you do find some kind of active genes which are heavily condensed, appears like condensed form. One of the classical example is nucleolus. Okay, within this particular nucleus, and then as I said, in humans we have 23 pairs of chromosomes, 23 pairs of DNA molecules, and if I ask where the DNA belongs to, every chromosome is seen within this intravascular nucleus. And then interestingly or surprisingly, or now we establish fact that DNA belongs to each and every chromosome within the interface nucleus, exists as a it's not diffused throughout the nucleus. It's seen as some kind of a territorial organization, and then they're restricted to particular location within the three-dimensional space of the nucleus. On this kind of constrained location of a particular chromosomal specific DNA during interface of nucleus is called chromosome territories. You always see a given chromosome territory, for example, chromosome 1, 2, and then other chromosome also two signals, because one chromosome could be mother chromosome, other chromosome could be from father chromosome. But always these homologous chromosomes are seen away from each other, as if they're enemies within the eukaryotic cell nucleus. So here the chromosomes traces, and then what is interesting is that when people have studied the location of a chromosome territories in multiple cell types with the help of a technique called spectral karyotyping, whereby which you can locate all the chromosomes of a particular cell type in a single cell by appropriately labeling different chromosomal DNA, and then from that, what the conclusion is that e- uh, chromosome territory positioning and then its neighborhood is cell type specific and is linked with cell type specific gene expression pattern. That means although all cells are having identical genome, but the way the DNA or chromosomal programs are positioned within the interfacial nucleus is unique for each and every cell type, suggesting that the way the DNA is packaged, the arranged in a cell type will actually dictate which genes are going to be transcriptionally active. And then further in strengthening the notion that there is cell type specific chromosome territory organization. So when people have scientists have studied the location of chromosome territories in a most fundamental cell, that's embryonic stem cell, and then which subsequently differentiated into intermediate cell and then subsequently differentiated somatic cell. When they study the chromosome territory positioning among these different stages of development, they learned that the chromosome territory positioning within the embryonic stem cell is highly random. Whereas as the cells gradually progress towards the differentiation, their chromosome territory positions are more or less fixed. That means more random nature of chromosome territory positioning from embryonic stem cell, gradually they go to non-random organization of chromosome territory positioning in a differentiated cell. And then this positioning again is unique for each and every cell type. Uh, Embryonic stem cell is being a pluripotent, so every chromosome is highly randomly placed within the new cell nucleus, and then as and when appropriate signals are received for a particular cell type, and then these chromosome territory organization seems to be reorganized, and then they are more or less more or less stable, and then they are non-randomly organized, and then where you see that unique transcription chromosome territory positioning and the neighborhood in a cell type specific manner. Further, this kind of chromosome neighborhood will allow chromosomes to interact extensively in a differentiated cell. That is chromosome interactions, which I will explain in my subsequent classes, has a lot of functional significance in terms of genes from different chromosomes and then talking to each other for a coordinated transcriptional control. Okay, so further now with the advent of molecular tools such as chromosome conformation capture and then microscopy in terms of confocal microscopy and then super resolution microscopy, now we know that within the chromosome territories how actually the chromatin is organized. So within the chromosome territory, you see that there are active regions and then there are silent regions. And then just for arbitrary sake, they have named it as a active chromatin regions are called compartment A and then compartment B. If you go deep into each compartment, you find such a tiny ball-like structure is nothing but these are the chromatin regions which are self-interacting and then form some kind of globular structures. These are called topologically associated domains. Now, today we know that topologically associated domains are the functional unit of eukaryotic chromatin. We know that nucleosomes are the structural unit of eukaryotic chromatin. These tags are now, we know that these are functional unit of chromatin, where within the tags you see that a set of genes and the regulatory elements, such as enhancers or silencers or insulator, they are seen within these particular tiny structures, which are called tags. Okay, so now by combining all the information that what we discussed so far in the electron microscope pictures, we have seen the condensed heterochromatin nuclear periphery, decondensed euchromatin at the nuclear interior, and then these condensed chromatin at the nuclear periphery associated with some kind of proteins called lamine group of proteins, and then lamine group of proteins they can bind to chromatin, and then if you wanted to better understanding, if you say single chromosome territory, if you imagine within the interface cell nucleus, you see that all the heterochromatic component or all the regions of the chromatin which are interspersed all along the length of the chromosome, so they associate with this lamine group of proteins and then they aggregate together in a long distance fashion, they condense and then they are pushed towards the nuclear periphery, whereas the euchromatin part, that means regions of the chromatin which are not in association with the lamine group of proteins, they are seen at the nuclear interior, and then this particular chromatin is highly loosely arranged so that any gene that is present in the euchromatic region, now they can access the DNA binding factors that are available within the cell nucleus. Okay, so within this particular loops, that what you see here, very broad loops, you see that, for example, here I have given you one locus where there is a long stretch of DNA where it is organized in the form of a loop formation, and then here there are four loops. In these four loops, that loop 2A has one regulatory element, that is enhancer, and then one gene, whereas a loop 2B has a single gene. In this kind of configuration, loop 2A has a gene and then promoter, because that now that enhancer can access this particular gene, the 2A specific gene can be transcriptionally active. Whereas a gene which is present, although it is present very adjacent to the this particular gene in terms of linear scale of DNA, but because of this loop organization, the gene that is present in the loop 2B cannot access this enhancer. Thus, these topologically associated domains or these loop organizations are independently regulated. That means the genes and then regulate elements that are present in a single loop, that are independently regulated. Thus, the adjacent loops are refractory to this kind of regulatory events. But in some instances, for example, in case of cancers, because of some kind of region, those proteins which forms this base of the loops, they are disturbed. As a result, these loops are not formed and it becomes a bigger loop. As you can see this, for example, in this example, gene otherwise which is silent, now it can access the enhancer because they are organized in a single loop. Now just imagine if it happens to be a proton gene which was kept silent in to originally to start with, and because of this kind of modifications, the loop organization is disturbed. As a result, now enhancer can access the otherwise silent gene, thus activating the oncogenes. Thus, that cell can now become a cancerous cell. That means the structural organization of genes not only dictates the cell type specific gene expression pattern, any alterations in the higher order chromatin organization can lead to dysregulation of gene expression, which we know that is a reminiscent of any kind of disease state. Okay, further, genes in a particular cell type or during differentiation process. For example, in a differentiation system where embryonic stem cells are programmed to become a neural progenitor cells and then astrocytes, and if you see that the heterochromatin region or the LAD regions which are seen as the nuclear periphery, they are very, very dynamic in nature. For example, in embryonic stem cell, the embryonic stem cell specific genes are seen away from the nuclear periphery. Thus, they're actively engaged in transcription process. Thus, these embryonic stem cell specific genes and then products will take care of embryonic stem cell state. Where the same embryonic stem cell, when it is programmed to become neural progenitor cell, all those embryonic stem cell specific genes are no more required Now they are heterochromatinized and they become part of the lamina, whereas the neuronal specific genes, which are otherwise a part of the lamina in embryonic stem cell, once they're when they're committed to become neural progenitor cell, as you can see that these chromatin regions which harbors neural specific genes, they are looped away from the nuclear lamina. Thus, this particular chromatin now they can access the transcription machinery for transcription activation, thus specifically activating the neural specific genes. And then for subsequently, when it become astrocytes, then astrocyte specific genes are specifically transcribed, whereas the genes which are not required for that particular cell type, they become the part of the lambda. So that means chromatin is dynamically moves from nuclear interior to nuclear periphery, or if this compartment A to B or B to A, thus will ensure that only genes that are required for to be transcribed, they are highly accessible, and rest of the genes are actually kept under silent by means of heterochromatinization. Okay, here I will stop. I think in my second part, I will discuss more about functional organization of eukaryotic cell nucleus. Thank you.